Hi everyone, we are Group 6 and we are here to present our topic, which is applications of bioinformatics tools for gut microbiome analysis.
Hello everyone, today I want to give a brief introduction to bioinformatics. It is an interdisciplinary subject that combines biology, computers, and mathematics. It uses computational methods to process and analyze large amounts of biological data, such as genes, RNA, and proteins, to help us better understand how life works. In the era of big data, many life science research are inseparable from the support of biometrics. It has become a very important and promising discipline. Thank you.
Uh, so starting with sequencing methods used in gut microbiome analysis, the three main methods are 16S RNA sequencing, whole genome sequencing, and metagenomics. Now, 16S RNA sequencing targets a conserved region of the bacterial ribosomal RNA gene, allowing us to identify bacterial species present in the sample. It's cost-effective and provides insights into bacterial composition. Whole genome sequencing (WGS) takes a broader approach by sequencing all DNA in the sample, capturing bacteria, viruses, fungi, and other microorganisms. This gives a more complete picture of the microbiome, revealing deeper insights into its role in health and disease. Metagenomics goes further by including both RNA and DNA. It allows us to analyze microbial functions and gene expression, showing not only which microbes are present, but also what they're doing in the body, such as helping with digestion or influencing disease. Once sequencing is complete, we need to ensure that the data is clean. This is where pre-processing comes in. We filter out low-quality sequences and contaminants and trim short or irrelevant sequences to focus on high-quality data. For taxonomic classification, we use tools like QIIME2 to identify which microbial species are present by comparing our sequences to reference databases. This step gives us a snapshot of the microbial community in the sample. Next, we perform functional profiling to understand what the microbes are doing. Human N2 maps microbial genes to metabolic pathways, revealing how microbes contribute to processes like digestion or metabolism. PICRUST depicts microbial functions based on 16S tRNA data, helping us identify functions like antibiotic resistance or enzyme activity, even without full genomic sequences.
So now let's look at some of the databases that can be used for gut microbiome analysis. First, we have Green Genes, a curated database of 16S rRNA gene sequences. What is 16S rRNA, you might ask? Well, it's one of specific gene that is present in all bacteria and is highly conserved, which is why scientists use it as a universal marker. Therefore, we can say that Green Genes is useful for identifying bacterial genetic sequences. It was once a popular database for storing bacterial sequences, however, it lacks recent updates and might not be used frequently today. Instead, researchers have switched to more newer databases such as Silva, which is basically a larger database than Green Genes, covers not only 16S but also 18S and 23S rRNA of bacteria and also eukaryotes. It also has more data formats stored, such as the ARB and EMBL formats. So here is a picture of the homepage of Silva. Uh, we also have another interesting database called the Human Microbiome Project. This was a major research initiative by the US National Institute of Health to collect microbiome data from healthy individuals. One of the sole purposes of HMP was to integrate reference data for normal composition of microbes in the human body and gut, which can then be used in comparative analysis of healthy against disease patients. Now let's discuss some potential tools that can be used alongside the databases mentioned for gut microbiome analysis. First, we have KIME2, which stands for Quantitative Insights into Microbioecology 2. It analyzes and visualizes microbiome data and is tailored more specifically for 16S RNA sequences. Some key features include processing raw sequencing data from gut samples, identifying the microbes present, and providing interactive visualization. It works well with Green Genes and Silva. Next, we have DADA2, which stands for Divisive Amplicon Denoising Algorithm 2. This is an effective tool in analyzing 16S RNA sequences because, as the name suggests, it corrects sequencing errors. One benefit of DADA2 is that it gives highly accurate sequences in the form of amplicon sequence variants, ASVs, instead of OTUs. OTUs stand for Operational Taxonomic Unit, as they group similar sequences together and based on a similarity threshold. ASVs basically take it a step further in accuracy. Lastly, we have Megan, which stands for Metagenome Analyzer. This tool is useful in analyzing large, complex metagenomic datasets. It's particularly useful in something called shotgun sequencing, which is randomly sequencing all the DNA in a sample. Due to its capability of analyzing large datasets, it pairs well with the HMP database.
Now we can look at four major clinical and real world applications of gut microbiome bioinformatics. The first one is in disease diagnosis and risk prediction. where bioinformatics tools like QIIME2 analyze microbiome data to identify disease-associated microbes. For example, high levels of Fusobacterium nucleatum in stool samples are linked to early-stage colorectal cancer. These microbial signatures can serve as a non-invasive markers for conditions like IBD and cancer. Second is in personalized nutrition and therapeutics, where platforms like Agora and Human N3 help understand how an individual's gut microbiota influences metabolism. Companies like Day2 use this data to offer AI-based diet plans that manage glucose levels. Bioinformatics also ensures safe donor selection for fecal microbiota transplants used in treating the species C. difficile infection. Third, we have in drug response and microbial drug discovery, where the microbiome's influence on how we process medications is increasingly clear. Tools like anti-SMASH help identify bacterial gene clusters that produce drug-like compounds. Studies show some gut bacteria improve cancer immunotherapy outcomes by supporting immune function. Lastly, we have the clinical trials and microbiome monitoring, where bioinformatics tracks how the gut microbiome changes over time in response to interventions like probiotics or dietary changes. QIIME2 is commonly used in these trials to analyze microbial diversity and correlate it with patient outcomes. Together, these applications show how bioinformatics turns gut microbiome data into powerful clinical tools for diagnosis, therapy, and precision.
Let's begin with the first case study conducted by Wang in 2021, which focused on the gut microbiome in HIV-1 infected individuals who were undergoing antiretroviral therapy. In this study, the researchers used whole genome shotgun metagenomic sequencing to compare the gut microbiome of HIV-positive individuals with healthy controls. The primary bioinformatics tools used was Kraken-2, which allowed them to perform high-resolution species-level taxonomic profiling. Their findings revealed a significant reduction in microbial diversity among the HIV group, along with an overrepresentation of specific bacteria such as Streptococcus aeruginosus and Actinomyces. More importantly, they identified the presence of resistant genes like APH3LA, which were positively correlated with longer durations of antiretroviral therapy. This suggested that the therapy might influence microbial resistant profiles over time. This case highlights the power of bioinformatics not only to map microbial communities but also to link them to clinical variables such as treatment response and immune system status. The second case study, published in 2024 by Kim Ma and colleagues, took a genome-resolved metagenomics approach to analyze the human gut microbiome. Rather than just identifying species, the researchers reconstructed metagenome assembled genomes or MAGs directly from raw sequencing data. This was done using a powerful bioinformatics pipeline called MetaRep, which integrates tools for read assembly, binding, quality control, and genome annotations. What makes this method special is its ability to assign functional genes, like those involved in metabolism or virulence, back to specific microbial genomes. This allows researchers to study microbes that have never been cultured in the lab. From over 110,000 of gut microbiome samples, they recovered thousands of high-quality genomes, many of which belongs to previously unknown species. This dataset contributes to what's now called the Human Reference Gut Microbiome, a growing global catalog of gut microbes. This study demonstrates of how advanced bioinformatics pipelines are pushing the boundaries of microbial discovery.
In conclusion, bioinformatics is now very essential in gut microbiome studies because of its capability to analyze microbial composition and function with tools such as QIIME2, Human N2, and MetaRap. It incorporates construction databases for taxonomic and functional profiling like SILVA, alongside 16S RNA sequencing and metagenomics, which ensure a precise evolution. In terms of the real world applications, it can be used for disease diagnosis, personalized nutrition, or even predicting reaction to drugs, which demonstrate the importance of microbiome research from a clinical perspective. Lastly, recent case studies have also shown that precision medicine can be developed through genome resolved methods understanding better microbial functions in human health and disease. These are our references. Thank you for listening.